You know, when a new disease emerges, the very first battle isn't fought with medicine, but with information. I mean, how do you fight an enemy you can't even see, let alone understand? Today we're diving into a revolutionary technology that's doing just that. It's called next-generation sequencing, or NGS for short. And believe me, it's giving us the power to decode and defeat outbreaks before they can spiral out of control.
So picture this. Alarms are basically going off in a public health office somewhere. A mysterious illness is popping up, and nobody knows what it is, where it's from, or how to stop it. Every single minute counts. And in this race against time, what's the biggest challenge? It's identifying the threat. To fight an invisible enemy, you first have to read its blueprint.
So to really get a grip on this, we're going to follow the journey of a single patient sample. We'll start at the very beginning of an outbreak, follow that sample into the lab, see how scientists piece together a pathogen's genetic puzzle, decode what that puzzle tells us, and then, finally, we'll look ahead to the future of disease surveillance.
All right, so our story kicks off in the middle of the chaos of a new outbreak. And the mission? It's brutally simple, but also incredibly urgent. Identify the pathogen causing the illness and figure out how to stop its spread. Fast. This, right here, is the high-stakes problem that NGS was built to solve.
Okay, so what is next-generation sequencing, really? Well, forget your standard microscope that just shows you the shape of a virus. That's old news. Think of NGS as a super high-speed reader for an organism's entire genetic instruction manual. It deciphers its DNA or RNA, letter by letter, with a speed that's just breathtaking, giving us a level of detail that was pretty much science fiction just a couple of decades ago.
Now, let's follow our patient's sample. It could be a bit of blood, or maybe just a simple swab, as it arrives at the lab. This is where the real scientific detective work kicks into high gear, transforming a biological fluid into pure, actionable digital information.
In the lab, that sample goes through 4 key stages. First, acquisition. Okay, the lab gets the sample. That's step 1. Easy enough. Next up, nucleic acid extraction. It sounds complicated, but it just means they're pulling out the raw genetic stuff, the DNA or RNA from everything else. Third is library preparation, and this part is just genius. They chop the DNA into tiny pieces and then attach a unique molecular barcode to each one. It's kind of like taking a book, shredding it into tiny pieces, but, and this is the clever part, putting a unique little address label on every single shred. Finally, the sequencer reads millions of these barcoded fragments all at once.
Okay, and right here, a really high-stakes choice has to be made, because not all sequencers are the same. This isn't just some technical decision. It's a strategic one that can have life-or-death consequences. Does the mission need the almost perfect accuracy of a big lab, or does it need the raw speed of a device you can use out in a remote field clinic?
And this table just lays out the trade-offs perfectly. So on one hand, you've got the aluminum iSeq. Think of it as the lab's workhorse. It's incredibly accurate, the gold standard, but it can take days. On the other hand, you have the Oxford Nanopore Minion, which is literally the size of a flash drive. It's portable, it can give you an answer in minutes, but the error rate is higher. So you see the dilemma, right? What do you need more in that first critical 24 hours? A perfect answer tomorrow or a pretty good answer right now?
All right, so the sequencing machine has done its job, but here's the thing. It doesn't spit out a nice, clean, complete genome. What you get is a massive data file with millions upon millions of tiny, jumbled genetic fragments. Now, the bioinformatics puzzle really begins.
I mean, it's seriously like taking 1,000 copies of the same book, running them all through a shredder, mixing up all the confetti, and then trying to piece one perfect copy of that book back together. That is the incredible challenge of computational biology, assembling that massive genetic puzzle from millions of tiny reads.
So how on earth do they solve this puzzle? Well, they've got 2 main strategies. The first is reference-based assembly. Think of it this way. It's like putting together a jigsaw puzzle when you have the finished picture on the box lid to guide you. You match your genetic fragments to a known genome of a similar virus. It's fast, it's accurate. But what if it's something totally new? That's where de novo assembly comes in. This is like solving the puzzle with no picture on the box, at all. The software has to figure out how the pieces overlap on its own. It's way, way harder, but it is absolutely essential for discovering a brand new threat.
Okay, so the puzzle is finally solved. The genetic code is assembled. We now have the pathogen's entire instruction manual. And this, right here, is the payoff. This is the moment it all clicks into place, where all that work translates into actionable intelligence that helps people on the ground.
And what this tiny little genome reveals, I mean, it's just astounding. You can finally identify the pathogen for sure. You can track its transmission from person to person with pinpoint accuracy, literally creating a family tree of the virus. You can screen for mutations that might make it resistant to drugs or even help it evade our vaccines. And most critically, you can spot something completely new. This is how data becomes life-saving action.
And we're not talking theory here. The speed in a real crisis can be absolutely staggering. During the West African Ebola outbreak, teams were using these portable sequencers in the field and got results in under 24 hours. The actual sequencing run itself, reading the virus's code, sometimes took as little as 15 minutes. 15 minutes! That is real-time genomic surveillance, right in the heart of an epidemic.
So that brings us to our final stop on this journey, the future. As powerful as this tech is, we are really just at the beginning of what it can do. So what's next? What does the next frontier in this fight even look like?
Well, first, let's get real for a second. This stuff isn't easy to just roll out everywhere. The machines, the chemicals, they're expensive. Analyzing the massive data files requires highly specialized expertise, and just storing all that information is a huge logistical challenge. You can see how these are major hurdles, especially for labs in places with limited resources.
But the future is also incredibly exciting. And the coolest part? It's becoming portable. The Zebra project, during the Zika outbreak, is the perfect example of this in action. Scientists took those backpack-sized MinION sequencers directly into Brazil, tracking the virus in real time. They didn't wait for samples to be shipped hundreds of miles away. The lab came directly to the outbreak.
And that just leaves us with one final, and I think really powerful question. As this technology gets smaller, faster, and cheaper, we are heading towards a future where a genomic sequencing lab can fit inside a backpack. In that world, could we actually spot and stamp out the next big pandemic before it even has a chance to start? That possibility, it's no longer science fiction. It's now within our grasp.